Hello, my dear students. My name is Sachin Jadhav, founder Fundamental Pharmacy, author of Fundamentals of GPAT Niper DI Pharmacist, Volume 1, GPAT Cracker Achiever MCQ book, and few other books. Welcomes you in this YouTube video session. If you are new on our channel, please subscribe the channel and press the bell icon so that you will never miss any video from me. My dear students, in this video, we are going to see pharmacology pinpoints, and particularly we are going to see the pinpoints from different phases of clinical trials and drug discovery. And a lot of times they have already asked the question on this point in a various kind of exam like GPAT, Niper, Drug Inspector and Government Pharmacist exam. So it's my request to you, please watch this video until end because this video is going to be very useful, knowledge oriented for these exams if you are preparing for these exams. So please watch this video until end. First of all, you should know what are the different processes for drug discovery. So drug is discovered by different methods. First method is a random screening, or it is also known as a high throughput screening. Random screening or high throughput screening. Now what you have to do in this method, you have to check the biological activity of different chemical structures. So you have different chemical structure or different chemical moieties or different chemical entities. Now you have to check the biological activity, whether this particular compound has a, or whether this particular structure has a particular pharmacological activity, and this method is known as a random screening or high throughput screening. Now another method is there known as a compound oriented screening or molecular modification. Now friends, what you have to do in this method, modification of existing drug molecule or modification of existing drug structure and optimization of that so that you will get a new molecule. Okay, for example, modification of acetazolamide has developed thiazide diuretics. So acetazolamide is a diuretic known as a carbonic acid anhydrase. So by modification of the structure of this acetazolamide, so another structure was developed known as a thiazides. They are also a diuretic. So it is another method, compound oriented screening or molecular modification. Now another method, third method is nothing but the target oriented approach. Now in this method, molecular targets you have to identify. So what are the molecular targets for a particular element or particular disease? For example, so if you want to develop a new drug for hypertension, so what is the target for developing the new antihypertensive drug? So that is nothing but the RAAS. RAAS means what? Renin angiotensin aldosterone system. So it is a new target for anti-hypertensive drug, renin angiotensin aldosterone system. So by targeting this system, few drugs category has been developed. For example, angiotensin converting enzyme, angiotensin converting enzyme inhibitors. Example is like captopril, enalapril, lisinopril, fosinopril, etc. So all these are the angiotensin converting enzyme inhibitors. Another category has been developed by targeting this renin angiotensin aldosterone system known as another drug category has been developed by targeting this renin angiotensin aldosterone system, that is nothing but the angiotensin receptor blockers. For example, telmisartan, losartan, irbesartan, etc. etc. So all these are the sartans are the angiotensin receptor blockers. Now third category, third method to discover a drug is nothing but the rational drug design. Rational drug design. So in this method, by studying the biological mechanism and receptor structure, you have to develop a drug. For example, proton pump inhibitors and selective COX-2 inhibitors. Proton pump inhibitors like omeprazole, rabeprazole, dextromethorphan, pantoprazole, etc. and selective COX-2 inhibitors like celecoxib, refocoxib, etrocoxib. So all these drugs are developed by this rational drug design. So first of all, you have to understand the biochemical mechanism, type of receptor, and then you have to develop the drug by targeting that receptor. So proton pump inhibitors and selective COX-2 inhibitors, they are developed by rational drug design. These are the four methods to develop a new drug, to discover a new drug. Now whatever drug you have discovered, that is known as a lead molecule. So using all above approach, these approaches will give you a lead molecule, and the process of finding lead molecule is nothing but the lead binding. Now from this lead molecule, once you got a lead molecule, you can modify that lead molecule by substituting a different substituent. Okay, the process is known as a lead optimization for a particular activity. So the process is known as a lead optimization process. Right? Now once you got a particular molecule or particular new drug, okay, particular drug molecule or particular drug entity. Now you have to check it for biological activity first using animals. Okay, so you have to go for preclinical studies. Now what do you mean by preclinical studies? To know the pharmacology, to know the pharmacokinetics, pharmacodynamics, toxicology and therapeutic index of newly synthesized or newly discovered drug. So you have to study its pharmacology, kinetics, dynamics, toxicology, therapeutic index, etc., etc., by using animals. Okay, you cannot use newly synthesized drug directly on humans. So for to check its biological activity, you have to go for animal study, and that animal study is known as a preclinical studies. Now newly developed drug, whatever new synthesized drug or whatever newly discovered drug is there, so that drug is tested on animals first. They are tested on small animals like mice, rats and guinea pig. First they will be tested on the small animals like mice, rats and guinea pigs, and after that they will be tested on relatively large animals like cats, dogs and monkey. And after the promising result or whatever is desired result, after getting a desired result of the particular compound, now that compound can be evaluated in the humans. Okay, so in the preclinical studies, whatever newly designed or newly discovered compound is there, it is tested on the animals, known as a preclinical studies. Animals like small animals like mice or rat, guinea pigs, or large animal like cats, dogs and monkeys. If you get a promising result or desired result, that means desired activity, then only the drug can be used in the humans. But you have to do preclinical studies first. Now what is meant by clinical studies? And remember, for preclinical studies you have to get a permission from animal ethical committee. Now what is meant by clinical studies? So if you got a very good result, if you got a very promising result in the preclinical studies, and now that drug can be used in the humans. Now to use the drug in humans or to study that drug in humans, you need to file a application known as a investigational new drug application, IND. So to check the to evaluate the drug in a humans, you need to file one application known as a investigational new drug. Now this application is filed to either US FDA. If you are living in the US, then US FDA is the authority. And if you are want to conduct the clinical trials in the India, CDSCO, Central Drug Standard Control Organization, is the agency. So you have to file the IND application for CDSCO or either US FDA to get a permission for testing of that newly discovered drug in the humans. And if permission is granted, if you got a permission to check or to test the newly discovered drug in the humans, that testing is known as a clinical trials. Okay, that testing is known as a clinical trials. So clinical trials, they are conducted in a different phases like phase 1, phase 2, phase 3, phase 4, phase 0. These studies should be conducted according to the ICH Good Clinical Practices Guideline. ICH Good Clinical Practices Guideline. So whatever study you are doing in the humans, all the studies should be in accordance or in accordance or with the norms given by ICH GCP. ICH means International Conference on Harmonization, and GCP means Good Clinical Practices. So the clinical study, whatever clinical trials you are conducting, the pages should be in accordance with the ICH GCP guidelines. Remember over here, so page 1, 2 and 3, page 1, 2, 3, page 1st, 2nd, 3rd needs ethical clearance from ethical committee, and page 4 does not need or it is not necessary to get clearance from ethical committee. These are the 4 drug discovery method. Remember, they might ask you a question on this. And what do you mean by preclinical studies? What do you mean by clinical studies? Now the most important point is the different stages of clinical studies, different phase of clinical trials. This is very important point. Already they have asked at least 5 to 6 times question in the GPAT exam, Niper exam, other exams also. So what are the different phases of clinical trial? Now first is nothing but the, you can see over here I have listed all the phases of clinical trial. Now phase first, phase second, phase third, phase 4 and phase 0. Now we'll discuss it one by one. What is the phase first study? How many number of participants are there? What kind of participants? What kind of study is there? And what is the major why this study is done? What is the significance of this study? Okay. Then time span of the study and the expenses, etc., etc., success rate. Okay. First one is nothing but the phase first. Phase first is nothing but the first in human. That means you are using your experimental drug in the humans first time. So that is nothing but the phase first study. Now, what kind of subject you have to choose, or what kind of volunteers you have to choose? Number of volunteers, it is nothing but the 10 to 100 participants or 10 to 100 volunteers, and which should be healthy volunteers. And remember, no extreme age range. Means what you meant by extreme age range? Extreme age range means, for example, those patients which are having very small age, like children's, or those patients which are having very old age, like geriatric patient. So that means in the phase first you have not to include geriatric patient and pediatric patient. Remember, इसमें geriatric patient, pediatric patient use नहीं करना है. इसमें healthy volunteers use करना है 10 to 100. Okay. Now most of the time, usually healthy volunteers are used, but occasionally patient with advanced or rare disease like cancer, so they can be used. In the phase first, most of the time you have to use healthy volunteers. Okay. यहाँ पे healthy volunteers या healthy subject आपको use करना है, लेकिन किसी किसी कंडीशन में, जैसे disease with advanced condition or rare disease, for example cancer. So in case of cancer you can use patients, okay, instead of healthy volunteers in the phase first. Now what kind of study is done in the phase first? Open label study is done in the phase first. Open label study is in the phase first. Now what is meant by open label? What is meant by randomized study? What is meant by control study? What is meant by placebo control? So it is, if you want to read that, you can read from books. So that is beyond the scope of this lecture. What is the main aim of this phase first study? What is the main aim of phase first study? So phase first clinical study is done for safety and tolerability, to check the safety and tolerability of the drug, whether your experimental drug is being safe in the humans or not. उसका टॉक्सिसिटी कितना होता है, उसका मैक्सिमम टॉलरेबल डोज कितना है, कितना डोज तक आप उसको यूज कर सकते हो ह्यूमंस में, ये चेक करने के लिए, सेफ्टी चेक करने के लिए, टॉलरेबिलिटी चेक करने के लिए अपन फेज फर्स्ट स्टडी करते हैं. सो मेन एम ऑफ फेज फर्स्ट क्लिनिकल study is nothing but the safety and tolerability, to check the safety of drug and tolerability of drug, to know the maximum tolerable dose, MTD, to know the maximum tolerable dose. So remember this. What is the aim? To check the safety and tolerability of experimental drug, to check the, or to know the maximum tolerable dose. Now phase first study, phase 1 study extend up to 1 month to year, कुछ महीने से लेके एक साल तक उसका स्टडी का ड्यूरेशन होता है. सो ड्यूरेशन ऑफ फेज फर्स्ट स्टडी इज अराउंड मंथ्स टू ईयर. इट विल कॉस्ट यू अराउंड $10 million. Okay. Then what is the success rate? The success rate for phase first study is around 50%. So अगर आपने 100% कंसिडर किया तो उसमें के 50% चांसेस है कि आपको फेज फर्स्ट स्टडी सक्सेस हो जाए. Okay. Again I will try to revise it quickly. So in the phase first means what? Phase first means you are using experimental drug first time in the humans. You need to identify 10 to 100 participants which are healthy volunteers. Okay, not like a geriatric patient or pediatric patient. Normally homologous population use usually healthy volunteers, and occasionally you can use patient with advanced disease or rare disease like cancer. Open label kind of study is done. The main aim of this phase 1 study is the safety and tolerability, to check the safety and tolerability, to know the maximum tolerable dose, and it extend up to few months to 1 year. It will take around $10 million expense, with 50% success rate is there. This was regarding the phase first. Now phase second study, phase second of clinical trial. So phase second is first in patient or subject. So you are using your experimental drug फर्स्ट टाइम इन द पेशेंट्स और सब्जेक्ट. पेशेंट जो भी आपके जिसके भी कंडीशन के लिए आपने ड्रग डिस्कवर किया है, जैसे मान लीजिए कि आपने डायबिटीज के लिए डिस्कवर किया है, तो पहले फेज में जिसको डायबिटीज है ऐसे लोग नहीं लेना है, यहाँ पे नॉर्मली हेल्दी वॉलंटियर लेना है फेज फर्स्ट में। फेज सेकंड में क्या करना है कि यहाँ पे पेशेंट लेना है, होमोलोगस पेशेंट, मतलब एक पर्टिकुलर एज के पेशेंट जिनको डायबिटीज है ऐसे पेशेंट लेना है। इन द फेज सेकंड मींस फर्स्ट टाइम यू आर यूजिंग एक्सपेरिमेंटल ड्रग इन द पेशेंट और सब्जेक्ट। नाउ यू हैव टू टेक अराउंड 50 to 500 or subject or volunteers. Now for phase second 550 to 500 volunteers are needed. Okay. Now in this patient or subject they are receiving your experimental drug over here in the phase second. Now what kind of study is done in the phase second? Randomized and controlled, or it can be placebo controlled or blinded study. So forget about this, let's concentrate on this point. When next, what is the main aim of phase second study? So main aim of phase second study is nothing but to check the efficacy and dose ranging establishment. To check the efficacy and dose ranging establishment. आपने जो भी ड्रग डिस्कवर किया है ना, आपने जो भी ड्रग डिस्कवर किया है उसको, वो ड्रग को आपके फार्माकोलॉजिकल इफेक्ट है या नहीं, सो उसको इफिकेसी है या नहीं। सो टू चेक द इफिकेसी ऑफ ड्रग, टू चेक द इफिकेसी ऑफ ड्रग एंड टू चेक द dose ranging establishment. So this study is done phase second clinical trial, and it is also known as therapeutic exploitation or therapeutic exploration. Remember this phase second is known as therapeutic exploitation. Now in the phase second study is done for duration of 1 to 2 years, and it may cost you around $20 million. 30% success rate is there. Okay. Now let's come to the phase 3 of clinical trial. What is mean by phase 3? Phase 3 is known as a multi-site trial. Phase 3 is known as a multi-site trial. Means multi means different, site means location, trial. So this trial is taken at a different kind of locations. Okay, remember. So what kind of subject are choose in this study? In the phase third, few hundred to thousands participants they are selected. Means for example, take an example of 500 to 1000 or 500 to 2000 or 500 to 4000. Okay, so few hundred dash thousands of students, they are thousands of participants, they are selected for the phase 3 of clinical trials from different centers. Now which kind of subjects or which kind of participants are there in the phase 3? Now patients, subjects, they are receiving experimental drug over here again, but in this phase 3 patients will be not a homologous series, it will be heterologous. That means all kind of patients they are included in the phase 3. That means here pediatric patient or geriatric patient and normal patients, then normal patients also included. That means all kind of patients they are included in the phase 3. Patient receiving the experimental drug again here. So up to this point, phase first, phase second, phase 3. So your drug is experimental drug, it is not approved. Okay? Your drug is experimental drug. So in the phase 3, the patient like geriatric and pediatric they are included. What kind of study is done in the phase 3? Randomized, controlled, and it can be placebo or blinded. Then what is the main aim of this phase 3 of clinical trial? So to confirm the efficacy in larger population and therapeutic confirmation. So remember this phase second is known as a therapeutic exploitation, phase 3 is known as a therapeutic confirmation. Okay, कंफर्म करना है आपको ज्यादा पॉपुलेशन में कि आपके ड्रग को एक्टिविटी है या नहीं, ठीक है, ह्यूमंस में. So to confirm the efficacy in larger population, and it is also known as a therapeutic confirmation phase. Okay, remember. Now it will take around 3 to 5 years duration to complete this study, and it will cost around 50 to 100 millions US dollar. Then 20 to 50% success rate is there. So remember in the phase first you are using experimental drug in the healthy volunteers. In the phase second you are using experimental drug in the homologous patients. In the phase 3 you are using again experimental drug in a extreme range patient like pediatric, geriatric, then young patients, etc, etc. Okay, remember this phase study is done for safety and tolerability to check the maximum tolerable dose. Phase second is done for the to check the efficacy and dose ranging establishment, or also known as a therapeutic exploitation phase, and phase 3 is known as a therapeutic confirmation phase. Remember in these all three phases you are using experimental. Now let's come to the phase 4. Now phase 4 is known as a post-marketing surveillance. Phase 4 is known as a post-marketing surveillance. Post-marketing surveillance. Now post-marketing surveillance means what? So after approval of drug, you can formulate your drug formulation and send in the market. Now post-marketing, after marketing of your drug, so whatever surveillance, whatever you are checking the long-term adverse effect, that is nothing but the post-marketing surveillance. That study, that kind of study is conducted in the phase 4 of clinical trials. Remember, now in this phase 4, so what kind of subjects are there? So many thousands of participants or subjects they are included in this study. That means after the marketing of your product, lot of patients they are taking your medicine. So many thousands of participants they are using your approved drug. Now remember, in this phase 4 your drug is approved. Okay, up to phase 1, 2 and 3 you it was an experimental drug. Now in the phase 4, that means your drug is approved. Okay, so the patient in a large population they are using they are using approved drug. Okay, in the phase 4. Now again, what kind of population they are using your approved drug? So patient in the treatment with approved drug. Okay, again, kind of study is the open label study. And what is the main aim of phase 4? To check the adverse reaction, then drug compliance, drug interaction. To check the adverse reaction, drug compliance, drug interaction and long term adverse effect is the main aim of this phase 4, that is known as a post marketing surveillance. Now it has no fixed duration, it will not cost is also not given and success rate is also not given. Okay, now most of the books consist of these 4 pages, but nowadays recently another phase is added known as a phase 0. Phase 0 is also known as a microdosing studies, and this study is done in the healthy volunteers of small numbers like 10 to 20, 5 to 20. Well, small volunteers, small healthy volunteers, phase 0. Okay, now in as the name indicate, microdosing means what? So whatever dose is there in humans, so 1/100th dose of that, 1/100, 1/100th dose of human is used in this. For example, dose in the micro, and this study is done to know the pharmacokinetics of the drug, to know the pharmacokinetics of the drug. So this is the different phases of clinical trials, and it is a very important. See, they have already asked question on this point. How many number of participants they are used in the phase 2, phase 1, phase 3, phase. So they were in the last year also there was a question. Many thousands of participants are included in the one of the following study. So it was a phase 4, GPAT 2021. GPAT 2019, they asked the question. So few hundred to thousand participants are included in the following kind of study, phase 3. So what is the aim of phase 1? They have already asked the question on this. What is the aim of phase 2? They have already asked the question on this. Phase 3, phase 4. So all these charts are, this chart is very important, and you will not find this chart in the reference book as it is, because this is a modified chart. Okay, ये चार्ट जो है एज इट इज आपको किसी के भी बुक में नहीं मिलेगा, बिकॉज ये थोड़ा सा मॉडिफाइड चार्ट है, ये मैंने मॉडिफाई करके बनाया है. ओके, सो दे हैव ऑलरेडी आस्क्ड क्वेश्चन ऑन दिस चार्ट in GPAT 18, 19, 20, 21, 22. So you can see consistently they are asking one question from this chart. Now question may be either direct question, they may ask you direct question, or they may ask you match the following type of question. So in the such kind of question they will give you in the one column phase 1, phase 2, phase 3, phase 4, and in the second column they will either give you the number of participants, or they will either give you the main aim of study. Okay, so such kind of question they used to ask. Once they already asked the geriatric and pediatric patients, they are included in the one of the following type of study. So इसके ऊपर भी एक बार क्वेश्चन पूछा है, बहुत सारे टाइप के क्वेश्चन यहाँ पे आते हैं. Okay, so for students, if you want such kind of full lectures, you can join our GPAT Niper 2023 online course. Here you will get live lectures on Wednesday, Saturday, Sunday. Recorded lectures will be available in our application. Study material will be provided to you in the form of hard copy at your address. Online test series will also be there. All this included in the 8,000 rupees, valid for 1 year. Okay, you can contact me on these numbers, and we have very promising result from past few years. Okay, and if anybody wants these books also, whatever the content I am presenting in front of you, so that content is from my book, okay, Fundamentals of GPAT. So if you want that book also, you can contact me on my numbers. Thank you very much for listening me.